Okay, so this is chapter 20, section 3, and this is all about mapping your DNA, figuring out the order of bases, the A's, T's, C's, and G's. So the most ambitious mapping project to date has been the sequencing of the human genome. This began as the Human Genome Project in 1990 and was largely completed by 2003. You probably learned about this in regular biology. Scientists have also sequenced other genomes, the fruit fly, nematode, worms, and so on, of other organisms as well. And so it allows the comparison between organisms' DNA, more specifically because you know the order of the bases. So how this all started out with was what we call linkage mapping, and we've already talked about linkage mapping with one of our genetics chapters. So the first stage of mapping a large genome is constructing a linkage map of several thousand genetic markers that should be mapped throughout each chromosome. The order of markers and relative distances between them are based on recombination frequencies. Remember when we talked about that before? The recombination frequencies is the percent of crossing over. So just a quick little review from before. You have your homologous chromosomes. So here's your sister chromatids of one chromosome. And remember, if genes are linked, they're on the same chromosome. So let's say the big A and the big B, and then let's say you're heterozygous for those two genes, and the little a and the little b. So crossing overs can occur between these two genes, and let's say you can switch the two B's. So you can pass on then a big A and a little b, and a little a and a big B. And so the percent of crossing over is directly related to how far apart the genes are on the same chromosome. So remember, the farther apart the genes are, the more crossing over that occurs. So if you have a high percentage of crossing over, that equals farther apart on the chromosome, and vice versa. If there's a low percentage of crossing over, a low recombination frequency, that means the genes are closer together. And so what we can do is make a linkage map to figure out how far the A gene is from the B gene. If there's a higher crossing over between A and B, then, and let's say there's a less crossing over between C and A, then you can figure out that C is between A and B, and so on and so forth. So this is a linkage map, and we did this with genetics. So that's the very first part. It's just kind of mapping out where the genes are on the chromosome. It doesn't give you exactly what the genes are, exactly where they're at, but you can get close ideas first where they are on the chromosome in relationship to one another. And so that's what this picture here is showing you. There's these different ways to map a chromosome. So the first one is the genetic linkage map. So this is based on recombination frequencies. Or those crossing over. And so genetic markers, these are, we'll look at certain genes and so on. So like this gene and this gene, let's say, would have a higher recombination frequency than this gene and the one right next to it. Okay, so it gives you relative distance. So then the two other pieces we're going to talk about here, that is DNA sequencing, so taking small segments, cutting the DNA up into small pieces, and taking those small pieces and sequencing them. So how do we know the order, the G's, the A's, the C's, the T's, and so on? And then we take those fragments once we sequence them, and we overlap them to figure out the overall order of bases for a longer piece of DNA. So that's what the basis of the next little bit of discussion is. So we'll write down some things, and then I'll give some examples, and I have a couple of videos as well. So a physical map, that's this part up here. So cutting it into pieces and then overlap the segments. So a physical map is constructed by cutting a DNA molecule into many short fragments and arranging them in order by identifying the overlaps. And I'll go over some examples of that in just a second. Physical mapping gives the actual distance and base pairs between markers, all right? So between markers or sections of genes. That's number 2. Number 3 is the DNA sequencing. And the DNA sequencing, you take relatively short DNA fragments that can be sequenced by what we call the dideoxy chain termination method. Another way to say this is Sanger sequencing. Sanger is the guy, the scientist who came up with this. And so that's another name for that. So let's go over this here in just a second. So in this, you have the inclusion of special dideoxyribonucleotides. So this is a new word here. Deoxyribonucleotide is a regular nucleotide. Deoxyribo is referring to without oxygen. It's the ribose without oxygen. That's the deoxyribose sugar. And then you have for the nucleotides, you have the phosphate, and then you have your base. So a dideoxyribonucleotide is missing, the di means 2, so it's missing 2 oxygens. And so you mix this in the reaction mixture that ensures the fragments of various lengths will be synthesized. And that's what this picture here is illustrating, is how we go about getting different fragments. And so let's look here. If we have... What we're going to do is in this box here, we have everything we need to accomplish DNA replication. So we have a single DNA strand, so we've broken the hydrogen bonds. It's not double-stranded, so here's your template strand. And then you have your primer. That DNA polymerase, remember, can't add nucleotides unless there's a primer present. You have your enzyme DNA polymerase. And then you have your deoxyribonucleotides. These are your normal nucleotides. And so the difference between all the nucleotides, all of them have the 3 phosphates and the phosphate and then the sugar deoxyribose and your base, either an A, C, G, or T. All right, then you have your dideoxynucleotides. These guys, let's write here, these guys are missing 2 oxygens. So when added in the growing DNA strand, it stops DNA synthesis. It stops DNA replication. I'll put replication/synthesis. So that's the significance of having this extra oxygen, missing this extra oxygen, is that it does this. It stops DNA replication. Another important part of this is that these guys are also in the lab fluorescently tagged. So they're tagged in different colors. So I'm going to make this one here purple. And then the C, I'm going to make pink. I'm just going to put it out here and box them out in pink. The T, your book uses yellow for the T. Yellow is kind of hard to see, so I'll use orange. And then we have the GTP, the G is green. So they're fluorescently tagged different colors. That'll be important in just a minute. So what happens is, see how we have a test tube here? We put all of this stuff in this test tube. So you're mixing all the stuff needed for replication together. And so you have to imagine what's going on in this test tube, and that's what this box is showing you here. So, so what happens is, first of all, you have your primer. Notice the primer matches the first 4 bases. So the new DNA strand is going to grow from the 5 to the 3' direction. So it's going to grow in this direction. So the primer comes over here, has the TTGT. That matches up with the AACA. So your TTGT primer can bind to that. Then DNA polymerase can come and add here a C to go with this G, a G to go with this C, and so on and so forth. The thing is that every time, though, DNA polymerase adds a base, a nucleotide, it has 2 choices. It can add a normal nucleotide, or it can add a dideoxynucleotide. If it adds the normal nucleotide, it can continue on and add the next nucleotide and the next nucleotide and so on. But if it adds a dideoxy one, it stops. So let's look at what happens here one by one. So when we look at this, let's just look at this first strand here. So here's your template strand. Your primer is added. So here's your primer right here in blue. Your TTGT. Now that matches up, so from right here to right here is your primer. So your next base is a G. Alright, so then you have the DNA polymerase is going to add a C here. But remember, it can add a normal C or a dideoxy C. So if it adds a dideoxy C, the C was pink. And so if it adds a dideoxy C, replication is finished. And because the DNA polymerase cannot add any more to that dideoxy C, and that's it. So you'll have a strand that's 5 bases, 5 nucleotides long. Let's look at the next part here. What if it adds a normal C? So it adds the normal C, so now DNA polymerase can add the next base. So what's the next base after the G is a C, so therefore DNA polymerase is going to add a G next. So again, it has 2 choices up here. It can add a regular G or a dideoxy C. G. If it adds a a dideoxy G, which is, I made it in green here, so now it adds this dideoxy G here. It can't add any more bases, and now we've got a finished DNA segment, and that is 6 nucleotides long. But if we look at the next picture, what if it adds a regular G? So here's the regular G here. So it goes to add the next base. So the next base in your template is a T. So again, it's going to add an A, but it can add a regular A or a dideoxy A. So if it adds a dideoxy A, that's the purple, replication stops, and you have a dideoxy A at the end, and you have a segment that is 7 nucleotides long with a fluorescently purple labeled dideoxy A at the end. If it adds a regular A, here we have the regular A, and it goes on to the next base. So the next base is a T, so then you're going to have a second A. Again, a regular or a dideoxy A. If it adds the dideoxy A, you're going to have the purple tag here. If it adds the regular, it keeps on going and goes to the next base, which will be a G. If it adds the dideoxy G, you're going to have purple. If it adds the regular G, we're going to continue on and go to the next base. The next base is an A. If it adds a dideoxy T to that A, you're going to have the orange fluorescently tagged here. But if it adds the regular T, it's going to go to the next base, which on the template is a G, so it's going to add a C. And so that's going to be a pink dideoxy. And then if it adds a regular C, you're going to go to the next base. Let me move this here. You're going to go to the next base, which is a T. If it adds an A to go with that T, a dideoxy A, it's going to be a fluorescently tagged purple. And then last but not least, if it adds a regular A, it now can go to the last base, which is a C. It's going to add a G to go with that. If it's a dideoxy G, it is going to be fluorescently tagged green. And so so this is what we have here. So in this test tube, you're going to have these fragments of all these different lengths, because at some point that DNA polymerase will pick a dideoxy one in each one of these spots, and you'll have these different lengths. And of course, you'll have some that are built all the way to you with a regular G as well. So then what we do is we put it in a gel. So the gel works kind of like gel electrophoresis in that it separates things based upon size. So these guys, the DNA, that's right here, DNA is separated, separated by passage through a gel with shorter strands moving faster, just like in gel electrophoresis. So our direction of the movement is down. So you're going to put all of this and the top here is going to move through the gel and separate based upon length. And it's so specific in that it can separate by length by just one nucleotide, which is the differences between these. So you get this separation here. Then when they use a laser to detect these fluorescently tagged pieces of DNA, and then the detector detects the colors. And so what happens is that this one, what's going to be the one at the bottom? The one at the bottom is going to be the shortest fragment, and so this is going to be radioactively or fluorescently tagged pink because it has a C here, and the laser is going to detect that and print this out, and the C is going to have a pink color. And then the next fragment is going to be 6 long, and it's going to be fluorescently tagged green. So then the next one is going to have the green color. And so what does that tell us? It tells us that after our primer, so we know our primer was TTGT, that the next base is a C. All right, so that's what this is. The next base after the primer is a C. The next base after that, because it's one more fragment long, one more nucleotide long, is going to be a G. The next base after that is going to be an A, so this will be purple because it's going to be an A. It's just a little bit longer, so it's not going to move quite as far in the gel, and we have our A. The next fragment is just one nucleotide longer, so it doesn't move as far in the gel, and it's also an A, and our printout is going to show purple, which then will tell us that the next base is an A. And so we can continue doing that here in color coding. So the next base would be a G, and the printout would color that and show that. The next would be a T, and so on and so forth. All right, and so then we have our T. Oops, I think I put the wrong color. I think that was supposed to be orange. All right, and then so we continue that. I might as well finish it out here. The C is pink. C is pink. And A is purple. Next longest. And lastly, G is green. And so that's how we know the order of the bases. So we know the primer, and then we know after the primer we just go up the gel shortest to longest, and it tells us after the primer what the order of the bases are. And that's how we sequence that. And so they've used this in figuring out the human genome, all the DNA and sequencing in the whole entire human genome. So what I'm going to do next is show you a little video here. So here we go. That's about the Human Genome Project.
As represented by this huge stack of paper, the human genome contains more than 3 billion nucleotides, or DNA letters. The first stage of the public Human Genome Project focused on identifying marker sequences, or unique tags, shown here in yellow, at regular intervals throughout this book of life. Once enough sequences were tagged, various blocks of the genome were allocated to different academic centers for sequencing. To begin the sequencing process, several copies of a section of DNA, represented here as a page of text, are cleaved to produce smaller fragments. Although it looks fairly orderly, this step is small-scale shotgun, which creates numerous random fragments. Each fragment is sequenced. Then computer programs align the overlap between fragments to build up an entire page. Marker sequences, shown in yellow, help establish the order of pages in the Book of Life. This methodical process produced huge amounts of data that have been used to virtually reassemble our genome. However, there are gaps. Repeat sequences are common in the human genome, so repeats from entirely different chromosome regions may be erroneously joined together. It will take many years to detect mismatches caused by the repeat sequences. Some regions, especially near the centromeres, may never be fully finished.
Okay, so this gives you an idea of what's going on with the Human Genome Project. The Human Genome Project used sequencing and this overlapping to map the human genome out, and it was a public venture, and so that means universities and so on. It was publicly funded, were different universities were working on it, and so on. And so therefore the results were also made public. So if we go here to the rest of the notes here, linkage mapping, physical mapping, and DNA sequence represent the overarching strategy of the Human Genome Project. But at the same time that the Human Genome Project was going on, there was a private company, Celera Genomics actually, that started to work on the same thing. So, and they used something a little bit different. So, alternative approach to sequencing genomes starts with sequencing random DNA fragments. Computer programs then assemble overlapping short sequences into one continuous sequence. And so this is kind of what the video that you just looked at was trying to show you. And so you have the DNA in the Book of Life they talked about, the DNA from many copies of an entire chromosome. So the many copies is key here. So let's say copy number 1 of the chromosome and copy number 2. All right, so this is showing you one copy here. But you cut it up so that you have overlapping sequences. So what does that mean? Let's say we cut this one and we have the sequence A, and then let's say we cut it again, we have B, and we cut it again and we have sequence or fragment C. So then when you cut the second copy of it, so this is an identical copy, so all the DNA is identical, we don't cut it in the exact same spot. So we have fragment D here. And so notice that fragment D will overlap parts of A and part of B here. And then if we, let's say we have a cutting here and we make fragment E has an overlapping part of B and of C. And then F has an overlapping part of C. So what happens is we cut these and we clone the fragments in plasmid or phage vectors, so we get lots of copies of these fragments that are short enough to be sequenced. So you have, let's say, this is fragment A, this is fragment B, this is fragment D, C, D, E, and let's say F. All right, so we have these different fragments, and we can have multiple copies of these. So we have more than one B and more than one F and so on. I'm just going to do it only once here for simplicity's sake. So that's what the cloning part means, that we get lots of copies of those. Then we sequence each fragment. So we can sequence each fragment by that dideoxy chain termination method that we just talked about, with the fluorescently labeled dideoxynucleotides, or that Sanger sequencing. So you sequence each one of these fragments. So you know the sequence of the fragments, and then you can compare them, and they often use detailed computer software to do this, but they order the sequence into one overall sequence to see the overlap. So for instance, let's say that this is fragment A, here, and then this is fragment D, let's say. So then we have some overlap. So see how this matches this part here? So the overlap shows that then possibly we only know this sequence and we know this sequence, but because they overlap, we can put them together and figure out the overall sequence. And then this one has an overlap here and an overlap here, so they go together. So therefore, by doing the overlapping here, we can figure out the whole order of the nucleotides from the beginning of this short sequence to the end of this short sequence because of the way that they overlap. And so that is how the Human Genome Project worked. Craig Venter for Celera Genomics used a little bit of a different method. I'm just going to show you a different video that he did, just to show you the difference. It's very similar but has slightly different. I think I'm going to show you the video. Technological errors here. Let me try this again. Oh. Well, I may have to just play it from here. I tried it out before I got on with you guys, so I'll have to play it. I'm going to pause it here. Alright, I think I got it to work.
Shotgun sequencing is the method that was used by the Private Genome Project. Shotgun sequencing requires multiple copies of the genome, which are effectively blown up into millions of small fragments. Each fragment is then sequenced. The small fragments are assembled using an immense amount of computer power to match overlapping sections. The drawback of this method comes when dealing with repeat sequences. Often there is no way of knowing how long the repeat sequence is, or in which of the many different possible positions the fragments overlap. Even the incredibly powerful software used to shotgun sequence the human genome couldn't cope with this. So Celera, the private company which relied on this approach, had to use the public data to fill in the gaps left by the repeats.
Okay, so that's the shotgun approach. So the Human Genome Project took the chromosomes and did it this way, and did it very systematically this way. Whereas the private company did that shotgun approach where they kind of visually took the quote-unquote book of life and blew it up into the whole genome into small segments, and then did the same overlapping sequence as well. So two different ways to do the same thing, and eventually between the two of them they got the job done. All right, and so that is 20.4. So I will be posting 20.5 soon.